Welcome everyone to episode 4 of our Designing Intelligence series, the last talk this term. My name's Chanan. I'm running this series of talks at Warwick Artificial Intelligence. Warwick AI aims to inspire, educate and connect students with AI. This term we ran 2 talk series, led projects, taught machine learning and Python, had a careers fair, connected you with world-leading research, welcomed you to a summit. I've certainly learned a lot, and that's not all. Next term will be just as exciting, if not more. You can also apply today for an executive position. If you're interested in becoming head of challenges or treasurer, then you'll be glad to hear that we are recruiting. But be quick, the deadline is tomorrow. As head of challenges, you'll be creating AI challenges for our members while working with many others in the team, industry, marketing, and treasurer. And as treasurer, you'll be managing the finances of the society, obtaining industry sponsorships and funding. More information is on our website. So today we are joined by Martin Lotz, who will be talking to us about the protein folding problem. As some of you may know, protein folding is the subject of recent news, featuring a breakthrough from DeepMind. Martin Lotz is an associate professor of mathematics at the University of Warwick. He specializes in computational mathematics and has interests in geometry and topology, among others. He was awarded inaugural Best Paper Award for of the Journal of Information and Inference in 2015. Martin, thank you very much for joining us.
Okay, so thank you for this introduction, and I will now share my screen. Okay, so hello everybody and welcome to this talk on AI for protein folding, and thanks to the organizer, in particular to Chanan, and also to Marion and Ziad, who are working on a project on this and who got me into this topic. And they are certainly by now more knowledgeable than I am on this topic, so if you have any, so with respect to details, they might be able to give you some more detailed information. Okay, so let's begin. Yeah. Okay. Now, why can't I share my screen? Yeah, just a second, I'll share my... So, is that working? Okay, that's good. Am I visible? Okay. Can you see that? Okay, great, perfect. Okay, so let's begin. If you have been following the news lately, well, there wasn't much happening, but this might have come up. So DeepMind AI cracks 50-year-old problem of protein folding. Problem solves scientific problem in stunning advance for understanding machinery of life. Or the game has changed, AI triumphs as solving protein structures. Science. DeepMind solves 50-year-old grand challenge with protein folding AI. London AI lab claims breakthrough that could accelerate drug discovery. And here they stole the image from the Warwick AI Society. Researchers at DeepMind say they have solved the protein folding problem, a task that has bedeviled scientists for more than 50 years. One of biology's biggest mysteries, largely solved by AI, and so on. DeepMind claims major breakthrough in understanding proteins. Discovery may dramatically speed up discovery of new drugs. It will change everything. DeepMind's AI makes gigantic leap in solving protein structures, and so on. So this has been making headlines a few weeks ago, actually last week even. So this is from November 30, probably 2 weeks ago or so. And so if you have seen this, you may have been wondering what this is all about. And this is what this talk will be about. So so what happened was that DeepMind have been able to predict the structure of proteins from amino acid sequences to an accuracy of more than 90%. So that's what, and I will discuss what this means in a while, but actually what today's topic will be, so we will be talking mostly about a precursor of this program that already achieved remarkable accuracy. So this, of course, builds on this precursor, so it improves it and it adds some new technologies to it, but the essence is similar. So we will be talking mostly about some outdated technology, which is maybe 2 years old, which is in the world of machine learning, this is considered outdated. But so the new improvements, they haven't been published yet. So the details on this, so they're just being published at the moment. So hopefully, from the information that you get today, so you will be in a position that you can hopefully get into this topic and then start learning more once new information comes out. Okay, so let's begin with first things first, so with the basics. And so what are proteins? Well, so it has become fashionable these days to label everything as being rich in proteins, and so many will know proteins as nutritional supplements that contain the stuff that is in red meat and tuna, things like that. But from a molecular point of view, proteins are very large molecules, so-called macromolecules, and that are essentially ingredients to the functioning of a cell. So they play a role in pretty much everything that's important in a cell, and they're literally everywhere. So what you see here is a kind of artistic impression of the inside of a cell, so with the membranes and all of these tiny bits that look like popcorn that are floating around here. So they're proteins of different structures, different sizes. And so as you can see, they're pretty much everywhere. And they play some essential roles, for example, in metabolic reactions, in DNA replication. So they respond to stimuli, so cell structure, and transport of molecules, regulating the immune system, signal transduction, and so on. So they're essentially the building blocks or the workhorses of anything that's happening in the cell, in the bodies, okay? And they're also the target of many drugs. So many drugs, they're specifically designed to target or to take advantage of the structure of certain proteins that happen in the body, okay? And let's now dig a bit deeper about how proteins are constructed. Here you see some images here that show you certain spirals, so certain long chains that are kind of folded in certain funny ways. So these are ways of representing proteins, but on a more basic level, if you go back to their basic structure, so proteins are chains of amino acids. Okay, so what are amino acids? Well, let's dig a bit deeper into what proteins are made of. So essentially that's the same stuff as DNA. So amino acids are certain organic molecules with a particular structure. So they contain an amine part, so NH2. So that depends on the nitrogen atom there, what you have here. And so they have another part, which is then a carboxyl part. So this is a carbon atom with 2 oxygens and hydrogen here. Okay. And then they have another group, so which is not in this model. There it's denoted by R1 or R2, which are the residues. And they can be different. So they can be different molecules with different structures. So they're just generally written at, but what all amino acids have in common is the structure that you see here. So the structure that you see above the R. So what's there written as R1 and R2. So this can be different. And there are approximately 500 naturally occurring amino acids that are known. but about only 20 or 21 of them actually appear as building blocks of proteins. And how do they assemble to make proteins? Well, so if you take two of these amino acids, so they can join, so they can, so this one, so you take away a piece of, let's take away this one here maybe. So a piece of hydrogen from one of these, and you take an oxygen and a hydrogen away from this, so you get H2O, which is water. You join them here, and then, so now you got two of them together, okay, with their corresponding residues, okay, and so this is water, this is then two amino acids, and you can keep going, okay, and in this way, so you can then generate proteins and also lots of water. So if you were wondering where all the water comes from, so as you can see, so you can turn them around, so they can have different structures. In this case, well, this is on the case of only two of them, but usually you have thousands of them. And such a long sequence of amino acids, that's what a protein is, essentially. And now, okay, so as I mentioned before that there are 21, so until recently it was thought there were 20, but you could say 21 or 20 such amino acids that constitute a protein. And so one interpretation is simply if you disregard the three-dimensional structure of these, so you can assign an individual letter to each one of these. And then, so you can think of this as being an alphabet. So you have 21 letters, and you can combine them in any way you like, or in almost any way you like. And that gives you an amino acid sequence that represents a protein. Okay, so you can interpret really a protein as a word, like a long word or a long sentence, if you like, consisting of these letters. That's really all there is, and this is very easy then to deal with, and you can characterize different proteins by their amino acid sequences. But there's actually more to this. So the sequence of amino acids in a protein gives you a lot of information, but it's by far not all the information that you get. And so the function of proteins does not only depend on the sequence of amino acids, but also on how they fold in space. And here one differentiates between different types of structures. So you have the primary structure, which is essentially the linear structure. This is just a sequence of amino acids. Then you have the secondary structure. So this is the way that these spiral maybe or form in different ways. Then you have a tertiary structure and a quaternary structure. Okay. And so it is possible to determine the structure of molecules by different means. So you can usually determine them experimentally using, I think the most popular technique would be X-ray crystallography, where say 90% of proteins have their structure determined in this way. But you can also use NMR, so nuclear magnetic resonance imaging, or cryo-electron microscopy. So there are different experimental ways to determine the 3D structure of these molecules, and they're usually quite involved. So it's usually a bit of an effort to determine these kind of structures. Okay, and so I'll show you one more example. So this was taken from a site on protein structure. So this shows you different ways what different proteins can look like, or how they can be visualized, and how their structure can be visualized in different ways. I mean, this is quite interesting, really, to see. So the kind of different shapes that you can get out of this. So you see on the, so I flipped this image around so that it can be seen better. So you can see above there on the top right, there's the insect flight muscle. So 2W49 or the adenovirus hexon protein, and different virus proteins that have a certain form. Okay, so we have seen now what proteins are. So they're sequences of amino acids that fold in a three-dimensional way. Okay, But in how many ways can such a protein fold? Well, so the structure, and the question is also whether the structure is completely determined by the linear amino acid sequence. So if you give me a word consisting of all of these letters, can I tell you exactly how the shape will end up looking like? Or can there be different shapes? And there's something that's called Levinthal's paradox. So this was posed by Levinthal in 1969. And what he noticed was that the degrees of freedom in an unfolded amino acid chain, they can be extremely large. I mean, so you can simply think of so if you have thousands of amino acids in a sequence, so the number of possible combinations, possible ways in which this can fold, so this can be just ridiculously large. Okay, but so when proteins are created, so they are usually translated from RNA, which is in turn transcribed from DNA. So you have DNA, so this is the genetic code, and that generates a strand of RNA, and which then generates these proteins. And this is also, by the way, so another thing that has been in the news was mRNA vaccines. So for COVID, that's essentially also how they work. So they have pieces of RNA that generate proteins, that certain spike proteins that are then being attacked by the immune system, and so when these are generated, so back to our topic, so when these proteins are generated from a sequence of amino acids, they quickly fold into a 3D structure almost instantly. So in many cases, they almost instantly fold into this particular structure that's characteristic for them. But if we are given such a sequence, then there are just billions of ways in which these can possibly fold. Okay, so famous manifestation of this discrepancy is again, so this was what's called Levinthal's paradox. But so obviously in nature there are some potentials at work that motivate things to fold in the most optimal way, but which we don't know, or this is something that we would like to understand how this works. Okay, and so you might think that this is a situation that's not that far away from. So if you think of the games of chess or Go that have recently been solved also by DeepMind, so where also the number of possibilities that you have there is also astronomical. So there's no chance that you can simply do trial and error and search for all possible options, or to go through all the possibilities there. But still, somehow they manage to become really good at these games by using artificial intelligence. And so something similar is happening here too. So you have this astronomical number of possibilities, but somehow, so you can learn, or you can train a system to learn how this works. And this is what has been done, and this is the main topic of this talk here. Okay, so I'll just give an overview, but you will find links at the end, so where you can then find more details on that. Okay, so let's move on. Now, so what is CASP? That's the critical assessment of structure prediction. Okay, and much progress in recent years on this topic has been because of this challenge. And this competition has been going on since 1994, and the latest installment is CASP 14, so it's the 14th time this has been going on. And it was there that the breakthrough by DeepMind that was recently announced in the news, so that I showed at the very beginning, that this breakthrough was made at CASP 14. And today's talk concentrates on the framework that was used for CASP 13, that was 2 years ago. And again, so as I mentioned before, in machine learning techniques that were popular 2 years ago almost seem antiquated now, so things are developing so quickly. But in any case, so the data, so for this challenge, this is usually based on data from the protein database, so that contains a lot of information with more than 100,000 entries. 150,000 entries for a lot of proteins. And so what they would usually do, they would take certain proteins and determine the structure of these experimentally. And the goal is then to predict this structure simply from the sequences. And then, so once this structure has been predicted, it's being compared then to the experimentally determined structure. And then if you have a high coincidence, so that's measured by a certain score, and if there's a high coincidence, then, or if you have an accuracy of over 90%, then it is determined as being solved. So then you can say that you actually pretty accurately determined the 3D structure of these things. Okay, so, but what does it mean to determine the 3D structure? So now let's go a bit deeper into what we actually want to achieve here. So in a naive way, in the most naive way, one can see this as a map that takes a sequence of letters, like one of these large words or sentences, representing the linear structure of a protein, namely simply the list of amino acids, and it maps it to the 3D coordinates of each element in the sequence. So each element is given a point in space, okay? So you can think of this as R3 to the power of n, right? So like n copies of R3. And so a different way of characterizing the structure would be through the torsion angles, okay? So there are essentially three of them. So if you look at this, again, if you take this model that we had here, so there, and I think this is really best visualized when actually looking at a particular model. So as you can see, the degrees of freedom that you have here. You can rotate this one here, you can rotate this one. So it doesn't really matter if you rotate this and this one you can't rotate, and this one here. So you have these essentially these three angles per amino acid, and the same here. But so one of them, so you usually is considered fixed. So actually you only care about two of them. So one is 180 degrees, it appears. So we only consider these two here, and then so among all the possibilities that you have of all the structural possibilities that you have. So just think of this model as being now much larger, consisting of so thousands of these things. So how many ways can you experiment with this? And so the natural approach that, say, a physicist would take or a mathematician to kind of solve this would be to try to construct some kind of potential or some kind of function that you try to minimize. So this is how nature works, right? So it always tries to minimize energy. And so you try to find something so when you minimize this function, you get an optimal configuration. So that would be the ideal case, to find such a type of potential. But of course, I mean, this is in practice, this is not very easy. So otherwise we wouldn't be talking here. And that is essentially the big challenge here, okay? So another way of characterizing the 3D structure of these molecules of proteins would be by looking at the distance between any residues, right? So you have these two residues. So these are the parts that, so certain parts of the proteins, and so two residues are considered to be in contact. So if they're very close together, so and close together usually means less than 8 angstrom, which is an atomic measure of distance. Okay, so and the distance between any two elements in the sequence can be visualized using a certain heat map. Okay, so here you have, for example, 140 or so times 140. So this is from a certain paper of these amino acids, and it's symmetric because it's a distance matrix. And so you can see, okay, so these ones are, so here, on the diagonal entries, they're all the same. So every one of these amino acids is close to itself. But here you have some that are closer to others and others that are farther away. And this tells you information about how these things are structured. So once you have this distance matrix, so you can go on then to construct a potential that will. So when you minimize this gives you a good structure prediction. Okay, and so the question is now, so how can you determine these distances from? So that's maybe a first intermediate goal. So before determining the complete structure is to determine this distance matrix based on the simply on the sequence data that you get. And so one way here is to take advantage of what's called coevolution. Okay, so you look at so the idea here is that many proteins are related, so they have a similar structure in the sense that they are generated by DNA segments that share common recent ancestors, okay, that are evolutionarily related. And the sequences of such proteins may coincide over large chunks, so they may be almost the same, but they differ occasionally, and this can be due to mutations or due to other divergence. And one can compare amino acid sequences, so that come from proteins, or one can do the same thing also with DNA, using a method that's called multiple sequence alignment, or MSA, which is quite commonly used in biochemistry or in bioinformatics. So it's one of the standard techniques in bioinformatics. And the idea is that you take also a lot of these sequences, a lot of these words. By the way, so you can also do this with language, so you can use the same techniques with any sequence of letters, right? And the idea is to align them in a way that you get the most coincidence, right? So sometimes some of these will coincide in a lot of places, but then there might be some isolated differences, or there might be some entry missing here, or sometimes you have something that's like here you have a Q and there you have an R, for example, so something that mutates, that switches around. But you try to maximize the coincidence that you have, and that will give you a measure of how much these individual ones are related to each other. Okay, and then so what can you do from this? Well, so first of all, you can find out how these different proteins are somehow related, but so what you would normally do is, so you take a sequence that you're interested in, then you look for some other pieces of protein sequences that correspond to the same domain, that are similar to that. And then, so what you look for is then for spaces, for places where, so for example, where you have correlated changes, right? So for example, if you have something here in one of these columns, so the idea is now that at certain sites or entries, proteins, they mutate in the same way, so they change in the same way to something else, and then they're considered to be somewhat related, so they could be either directly correlated or it could be through a third site that both of them are close to. So for example, if this one switches to K always at the same time that this one switches to E, then you think that there's a certain correlation here, and this could be due to the two sites, these two positions being close together. So they could be like here, so they could be close together, which would imply that maybe changes to one part would lead to similar changes, would also lead to changes to the other part, and every time when one of these sites changes, the other one changes too. So that kind of can give you an indication that there's a certain distance between that is small, so that they interact in a way. So they're not independent dispositions. Well, for example, you could have other places in the sequence of words that are far away from each other and are quite independent. So something that happens to one of these positions when you get a mutation in one of them, so it could be completely unrelated to what you get in the other one. Okay, so and the idea is to relate this somehow to the distance in the three-dimensional structure that you want to have. Okay, so of course this is in reality this is all a bit more complicated. So ideally, what you want to get is from a certain sequence and then certain homologous sequences sequences or certain close sequences that you have. So you want to construct a kind of probability distribution on the individual letters of that sequence that tell you not only what the probability is of a certain letter from happening. For example, here you have you would count the frequency here. At this side, you would see that 5 of them are R and 2 of them are K. So there's a 2/7 probability, 2/7 probability of this being K. So you can determine certain probabilities of certain letters in a certain position from this, but you can also determine joint probabilities. Say the probability that this is K and this is E, both at the same time, and this way you get information about how these different positions are correlated. And so one way of formalizing this is using a certain concept from physics, which is called the Potts model. So you think again of a group of related proteins, so segments. So you have a sequence S1 to Sn, so you have n letters, like a word consisting of n letters. Okay, and then from the multiple sequence so alignment, then so you can then determine certain probabilities by looking at frequencies. For example, this would be the probability that at the ith position you have the letter K. Okay, and here is the probability that at the ith position you have K and the jth position you have L. Okay, and from this you can construct then a probability distribution on the whole letters, which is in what's called the Potts model. And so you can determine this using certain techniques like quasi-maximum likelihood estimation or some entropy maximization procedures. So we'll not go into the details, but the point is, so you get some kind of probabilistic model to describe what's happening in your sequence, and that you can also then exploit, hopefully, to make some predictions about the distances. So how does this work? Okay, so to summarize what we have seen so far, we take a sequence S, we take a multiple sequence alignment, which is, so we compare this with similar sequences from the database, and we extract some features from it. And then, so from this, we want to then infer something about the distances between any two sides in our sequence of letters. And the way this is done is so we can't really say exactly what the distance is, but we have certain probabilities, a certain probability distribution on these, and also a certain probability distribution on the torsion angles, ideally. Okay, so you want to have certain probabilities, say, so you want to be able to say that the probability that the torsion angles are in a certain way is high or is low if you change them, okay? And again, so the magic that happens in between, so that's the first point where neural networks come into play, and a particular type of neural network, what they call a deep dilated residual neural network. So this is a new type of neural networks that are being used here in this context, okay? And they, so how do they work? So let me briefly, before going a bit on to the next steps, so let me just briefly give a primer on, well, a refresher on how neural networks work. So I have been told that you already had some introductions to deep learning and neural networks. So here is a one-slide introduction to neural networks. So a neural network is essentially a function that maps inputs to outputs, but it's a function of a very special form. So it's a composite function. It's a function where you apply one function and the next, and so on, and the next. And each of these functions can be seen as representing a certain layer. And what happens at each layer? Well, you get a linear map. You get W times X plus B, where the W is a weight matrix and B is a bias term. But it would be boring if it would just be linear maps, because if you compose linear maps, you end up with a linear map. So to make things more interesting, you introduce some nonlinear functions, some activation functions in between, to go from one step to the next. And here are some typical examples. So this is the way a neural network is normally visualized. Okay, so these are the different layers, and these arrows represent the different weights. And these are three examples of commonly used activation functions, so the sigmoid, the hyperbolic tangent, or very popular, the ReLU, which is simply 0, and then it's just the identity. Okay. Okay, and so how do you determine the parameters of a neural network? Well, you want to use it for prediction, so you take some training data, you take some inputs xi, outputs yi, you have a loss function, which could be the square difference or the cross entropy or some other terms. And then, so you simply try to minimize this loss, so you try to find the best fit, or the weights W and the bias terms B that best fit this model. And the algorithm of choice here is stochastic gradient descent, which is a form of gradient descent that works for very large data sets. And the gradients can be computed again by an implementation of the chain rule, which is known as backpropagation. So if you recall that a neural network is a composition of functions, so if you want to compute gradients of that, then of course you use the chain rule. And there are also other methods that you can use for optimizing this. But the idea is, if you have training data, if you have inputs and outputs, and you have a loss function, for example, cross entropy, then you can determine optimal weights that fit this data, and you can then use this to make predictions. Okay. Okay. Now there are certain special types of neural networks, certain architectures, so to say. So one is what's called a convolutional neural network, which is used for image data quite a lot. So, and the linear maps there in one or more layers are called convolutions. So what they do is they usually take small parts of the image, input image, and then they apply a certain map to them called a convolution, and they do this for different chunks, and then there's a certain next layer that simply does subsampling. So it takes, so from 4 entries maybe, or from any 2x2 block, it just selects the maximum entry and places it here, and then it does again convolution, subsampling, and then it has some fully connected layers. So this would be the typical architecture that you might expect from a convolutional neural network, say for digit recognition, right? So this could be one that recognizes digits 0 to 9 or letters or whatever. Okay, and there are countless other examples of these kind of neural networks. But the problem with these is that usually, traditionally, it was thought that if you increase the number of layers, then the neural network becomes more powerful and can perform better. But it turns out that this is not necessarily the case. So there are certain problems, like what's called the vanishing gradient problem, which can be fixed, but there are also other problems that show that the performance can actually deteriorate if you make your network too deep. So if you take 50 layers instead of 10, then the performance can actually deteriorate. So one way out of this has been to use some kind of tricks, like and to use something that's been called residual neural networks. So that kind of add some modifications to the different layers. So for example, they can add to the output of a certain layer, they can add what comes from a previous layer, or a linear transformation of what comes from a previous layer. And it has been shown also experimentally that the performance of such neural networks tends to be better when you have very deep neural networks. And a very deep neural network is what has been used here in our case. So this is the residual neural network, or this is like a schematic representation. And I'm not sure, I mean, this is not very accurate what I'm showing you here. So depending on where you look, they give you different information, but that's not really the point. I mean, so the precise structure is, I mean, this can vary. One can experiment with using different types of structures, but this is more or less what has been used in AlphaFold to go from the step of having your amino acid sequences and their MSAs, the multiple sequence alignment, and features extracted from that, and to get from there to the distance matrices. Okay, and so here are some of the predictions that have been done in this way. So this is from their paper. It has been published in Nature only at the beginning of this year. So it shows some of the distance predictions. Here's the ground truth. So this is the true image that has been generated. So this is what has been predicted here, right? And here's and these pictures here, they show the coincidence between the experimentally, so the ground truth, the experimentally determined images, and the ones that have been predicted. Okay, so let's just summarize this whole procedure. So you have this this pipeline, roughly speaking. So for the torsion distribution and the distance distribution, and then you add some term, and then you create a potential here. So once you have them, you create this potential, and you can minimize this potential then using gradient descent, and that will give you an optimal configuration that you can then use to compare with the experimentally determined one. And here's the step where the residual neural network comes into play. So you don't worry about these. So these are algorithms for multiple sequence alignment that they generate certain features that get fed into the network, and it determines as output then the torsion distributions and the distance distributions. Okay, and okay, so once you have these, well, so once you have these probabilities, so you can form the potentials in this way. So you can form a potential for the location simply by taking the sum, essentially a log likelihood function, like the sum of the log probabilities that have been determined. And here, these are conditional probabilities on the sequence and the multiple sequence alignment that you get. And here you have the probability distribution of the individual torsion angles corresponding to the ith amino acid here. And then, so you try then to minimize these. So you try to find the optimal parameters. And so what this image shows, so this is from DeepMind, so it shows so what happens while this is being minimized and how gradient descent arrives at the optimal configuration of these potentials here. So what happens at every step, so you start with a linear sequence, and then eventually it settles into this 3D structure. And what you see here, this is like a toy example. So this is not it. So in reality, the molecules that they have been dealing with are much bigger than this. But this is simply an animation that shows where it's coming from. Okay, so we're coming to an end. The basic reference here is the Nature paper by DeepMind, and you can find more information. You can also find the code based on TensorFlow, and you can also find a free implementation based on PyTorch. So you can find all of that linked on the website by DeepMind, so where they discuss this. So a new version, so as I mentioned before, so the results that have been in the news 2 weeks ago or so, they refer to the new updated code by them, and it hasn't been fully, as far as I'm aware, so I haven't checked today, but it seems that it hasn't been fully published yet, even though there's information coming out on how this works. So the precise details on this updated version with increased accuracy are not yet available, but hopefully will be soon. And also the new version is based on, apparently, on some more advanced deep learning techniques or some new developments, so that include generative models and also certain new technologies. But the details, again, are not fully out yet. So there's an abstract available, and there's some analysis that I've seen, but this is certainly very exciting, and it's certainly, I would recommend, if you're familiar with using Python and TensorFlow or PyTorch, to experiment with this. So go get the code, go try this yourself, and then maybe try to, if you're interested in, maybe try to see whether you can recreate some of the results or have maybe different ideas on how to work on this. Okay, that would be it for now. And thank you very much. And if you have any questions, I'm around.
Okay. Camera's coming up. Cool. Thank you, Martin, for your insightful talk. There aren't any questions in the live chat, so if anyone does have any questions, please don't hesitate to write them in. So I have some questions. So firstly, what applications are there of accurate protein folding predictions beyond biology?
Oh, beyond biology. Well, I mean, so the application in biology, as far as I see, is that you can just speed up research by a lot. I mean, so you don't really have to go too costly, and so sometimes when you're interested in protein structures, and this is something that's already being used, so it kind of makes it faster and cheaper to determine the structure of proteins, right? So you can find drugs. Now, outside of biology, well, I think one of the benefits is like with the reinforcement learning that has been used for chess or Go, where also you can ask what's the use of this outside of these games. So I think it simply advances the technology. It can be used as a benchmark for new machine learning techniques. So it's like with a lot of technologies. So you work hard on I mean, even in pure mathematics, so if you solve a problem like Fermat's last theorem or so, to actually be able to solve it, you have to develop new technologies and new ideas, whole new theories, a new way of looking at things, and it's probably similar here. So these are simply simple problems that simply the fact that you can solve them means that you have made some advance in your understanding of what intelligence is and how you can so use it to solve problems.
Okay, so do you sort of see that advancements towards our understanding of intelligence comes hand in hand with advancements and applications of AI?
Yes, I think so. I mean, definitely. I mean, so it has been said that we understand now that we protein folding, but I mean, we don't really understand how the neural network does it. I mean, we train it, and essentially the machine that we train understands it, not we. But we kind of understand how to approach certain problems or how to solve them, maybe in a better way. But still, what's going on there might still be so it depends what you mean by understanding, really, in the end.
There's a question from Math Potato. How large was the training data set?
Oh, I'd have to check. It was, so I think they used like 3,000, 30, no, let's see, details. They split this into, I think I have it here. So they used a small test set, but the training set, yeah, it was 30,000 or so, around, slightly less, well, pretty much exactly 30,000 proteins. So for the training set, and for the test set around 2,000. Yeah. But one has to keep in mind that each of these sequences are very long, so they, yeah. So the dimension is large. The number of samples is also large, but I think the dimension is really different.
Okay, and so DeepMind's solved the problem, as CASP has announced. Is that the end of the 50-year-old problem, or is there more to be done?
Well, I think you can always increase the accuracy. I mean, so this thing with 90%, that's pretty arbitrary. I mean, so probably 90% is what people would consider to be good enough, maybe for practical purposes, I assume. I mean, I'm not a biologist or I'm not an expert in medical applications, but I think that's what people thought at the time when the challenge was devised, what they thought that they would be happy with. I mean, there's always room for improvement, certainly, and improvement can go in different directions. So it can be an improvement in accuracy, but I also see that there can be a lot of improvement in the efficiency and the simplicity of the methods. So it's quite conceivable that you can come up with much simpler structures and much easier ones. That it's again to compare this with chess or Go, where you also, as at the beginning, you had to train them, so using a lot of games, and then so in the end you ended up with a solution that kind of was able to learn the game just by playing against itself without any other data. And here also you can do certain improvements, for example, in trying to get rid of the multiple sequence alignments or a certain auxiliary data that you put into this, or auxiliary pre-processing that is going into this. So you can certainly improve the efficiency of the method itself.
And a question from Aman Kumar. He says, how has AlphaFold solved the protein folding problem? I think it's not quite clear to me what he means, but I think just if you could give an explanation of what it really means to solve such a big problem.
Well, so what it means is it's okay. Let me just go. It's well, they have a certain. So they're given certain examples from the challenge. I mean, they have to be able to predict most of them with a certain score. So it's simply achieving a certain accuracy, and that is being measured in a certain way. So it's I mean the details are a bit more complicated. For example, how do you measure? So how do you compare the predicted structure with the experimentally determined one? So there's something that they use, the TM score, which is a certain measure of similarity that they use, and they have certain benchmarks there. That so in a way, being solved is not really a discrete thing. So it's not that. But it's something that's incremental. So you increase the accuracy with which you make your predictions, and then once the accuracy given certain metrics is high enough, then you declare it to be solved. But it's not that that you simply win a game, or that you. So it's not something discrete. Yeah.
Okay, thank you. And
so I should add that the experimental methods are also not perfect in making these predictions. So you kind of compare it with the experimental method, but really the ground truth, that's something you don't know. So you want to get at least to the accuracy of the experimental methods, and so when you reach that accuracy, then you can say that it's solved. Yeah.
Okay. And a question from Adam. Does every input into AlphaFold require many sequences to calculate MSA?
Yes. Well, the way I understand it, yes. I mean, this is something that they, even though they yes. So kind of, you don't just put in the single sequence. Yeah. I mean, how many of them? So we'd have to check, but so I think so when you look at the code, so they have the so they already have the data prepared in some way, so the features that you can enter them. So